question is, why was the patient dead? Not the anatomy was wrong, something was missing. What was missing was energy. Newton said a half a millennia ago, mass doesn't move without energy. And you're the most animated thing in my environment. So therefore, being animated and being mass, energy must be critical. So why isn't half of medicine about energy? And yet, as far as I know, in most medical schools, you memorize the TCA cycle, go to sleep, and forget about it. Okay, so if life is about having anatomy to have structure and energy to be animated, then you need information about anatomy and information about energy. Well, if that's true, then maybe if we think about energy, maybe a lot of these diseases might be about energy. So our basic idea is that, in fact, energy deficiency is the basis of all the common complex diseases. And interestingly enough, the most important information about energy is not on the chromosomes. So therefore, it does not Mendelize. So if that's the case, then you might come up with a new hypothesis. Our hypothesis that we've been testing for the last 45 years is bioenergetic dysfunction lies at the nexus of genetic and environmental causes, and that the common complex diseases are bioenergetic diseases, not anatomical diseases. So this is your biochemistry lesson from hell. I've never met a medical student that liked this, and I love it, and I like torturing medical students, so we go through this. Okay, so this is a symbiotic bacteria. Sitting in your chair right now are 10 to the 12th living individuals. Those are called cells. And each one of them has an independent life. We can take a little sample of your skin, put it in culture. Cells will grow perfectly fine. You can go out and get run over by a truck. Your cells don't care. They're growing perfectly fine. So you are a colony of cells. The idea of an I is purely an illusion. You're a they. Okay, or a we, let's say. But inside each of the we, there's another colony of cells. And that colony of bacteria, the they, is in fact the most common cell in your body. There are 10 to the 17th of these bacteria sitting in your chair right now, in the order of 30% of your weight. And those bacteria are called the mitochondria. So they enter the cell about 2.5 billion years ago, and this was the outer phagocytic vesicle, if you will, and this is the original bacterial membrane. So this is just one of those bacteria. So this is the outer mitochondrial membrane, intermembrane space, intermitochondrial membrane, and the mitochondrial matrix. So this is, of course, as you've been told, the power plant of the cell, which is wholly simplistic. But you take carbohydrates and fats from your environment, you strip the hydrogens off those carbohydrates and fats, and react it with the oxygen that you're breathing, and you burn that to give water and generate energy. And if you don't think this lecture is important, just stop breathing for the next 10 minutes, and you can go. Okay. So let's say we're talking about glucose. Well, it goes through glycolysis to make pyruvate. Pyruvate can be reduced to lactate, or it can get an amino group to give you alanine. If there's a big block in the mitochondrial function, pyruvate goes through pyruvate dehydrogenase, which will generate acetyl-CoA, driving the tricarboxylic acid cycle, the purpose of which is not to anesthetize medical students, but to put hydrogen on this carrier, NAD. So here is the oxidized and the reduced form with the two hydrogens. Now, NADH is oxidized by NADH dehydrogenase, or complex 1. The electrons flow through complex 1, CoQ, complex 3, cytochrome C, cytochrome C oxidase, or complex 4, and reduce an atom of oxygen, give you a molecule of water. Now, if all you were doing was burning electrons, we'd just generate a lot of heat, and you would, of course, melt down. That wouldn't be good. So how do we get that energy to use it in a way that we want to use it for different purposes? Well, as the electrons flow through complex 1, 3, and 4, they use that energy to pump protons from the mitochondrial matrix through the lipid bilayer, which is an insulator, into the intermembrane space. And as Peter Mitchell hypothesized, then that energy is based on an electrochemical gradient. That's positive and acid on the outside, and alkaline and negative on the inside. And then he proposed that potential energy was used by the ATP synthase, for which one of your faculty members won the Nobel Prize, because as the protons move through the system, a spinning wheel causes rotation, which then causes binding of ADP and phosphate to condense ATP. And then the ATP goes out through the adenine nucleotide translocator, out through a sieve-like protein, the voltage-dependent anion channel. There, ATP is hydrolyzed to make ADP to do work. So you've coupled phosphorylation with oxidation, oxidative phosphorylation. Now, everybody in this room has a slightly different efficiency at pumping protons out versus calories burned and converting them to ATP. We call that coupling efficiency. Now, a calorie is a unit of heat. Every calorie that you burn generates that amount of heat inside your body. So, in fact, if you're very efficient at pumping protons out and converting them to ATP, then you'll have to burn the minimum number of hydrogen calories to make the maximum amount of ATP, and as a result, you'll generate the minimum amount of core body heat. However, if you're less efficient at pumping protons out or converting them to ATP, then you're going to have to burn more calories for the same amount of work, and that's going to generate more heat. So your body then is monitoring your body temperature at 37 degrees centigrade versus your workload based on the coupling efficiency. Okay? Now, it's a furnace, so it's going to have incomplete combustion, so electrons can go directly to O2 to give you an unpaired electron in the outer orbital, superoxide anion. Manganese SOD will take 2 of these to make hydrogen peroxide. Hydrogen peroxide could get another electron by a reduced transition metal to give you a hydroxyl radical, and these are the reactive oxygen species, or the oxygen radicals. So if you take vitamin E or vitamin C or beta-carotene, or coenzyme Q, you are concerned that you are getting oxidative damage from these oxygen radicals. Now, your cells have an enzyme, nicotinamide nucleoside transhydrogenase, that makes NADPH, and glutathione peroxidase that uses that NADPH to reduce hydrogen peroxide to water, and that then detoxifies the hydrogen peroxide. But this is a rate-limiting step, so there's an excess of this. The mitochondria also being a capacitor, it's negatively charged inside. It has the capacity to take up calcium, and it's the major calcium regulator in your cells, and that's important because calcium is the most important inorganic molecule or atom in regulating a lot of biochemical processes. And finally, it has a self-destruct system, the structure of which is actively debated, but normally it's a closed door. But when the energy potential declines, oxidative stress gets high, calcium overload becomes excessive, impinge on this pore, it goes from a closed door to an open channel. The open channel short circuits the membrane potential, fluids flow in, the inner membrane swells, backs and back form a megachannel. They release all these stored intermembrane proteins, including cytochrome C. They go out and they drive apoptosis and degrade the cell from inside out. So the mitochondria has lots of different functions. It regulates the energy. It regulates the redox balance, and about two-thirds of all the proteins are thiodisulfide regulated. It regulates reactive oxygen species, which are signaling molecules at low level are damaging at high levels. Regulates calcium, regulates apoptosis, and all these TCA cycle intermediates directly are the intermediates for all the enzymes that regulate the signal transduction system and the epigenome of the nucleus. Because, why? The nucleus can't do anything without enough energy. It has to know what the mitochondrial energetic state is, and therefore it uses mitochondrial metabolism to assess that. So your cell is 2 life forms, a nuclear cytosolic life form, which was originally a methanogen of the archaeobacterial form. It has DNA in the chromosomes, transcribed in RNA, translated onto cytosolic ribosomes, and between 1,000 and 2,000 anatomical proteins of the nucleus go into the mitochondria to assemble the mitochondrial structure. But the mitochondrial genome, which originally was co-equal with this genome, besides transferring all the anatomical genes to the nucleus, it's retained 13 polypeptides, 22 transfer RNAs, and a small and large ribosomal RNA. And those transfer RNAs and ribosomal RNAs make mitochondrial-specific ribosomes. Mitochondrial translation is initiated by N-formyl methionine, just like bacterial ribosomes. These ribosomes are sensitive to chloramphenicol and aminoglycosides, just like bacteria are, and it codes for 13 polypeptides. So what are those? There's 7 of the 45 proteins of complex 1, 1 of the 11 proteins of complex 3, 3 of the 13 proteins of complex 4, and 2 of the 17 proteins of complex 5. Well, if you're going to put 2,000 genes in the nucleus, why keep 13? The answer, I believe, is because those are the electron and proton carriers for these 4 enzymes. And what do these 4 enzymes share? They share the membrane potential. Now, this membrane potential is a capacitor. If one of these became leaky for protons, it would short the circuit and you would be dead. So Mother Nature was very concerned about that. She said, well, how am I going to balance the evolution of all of these enzymes so that they stay together? And so she had a great idea. Being a woman, she decided that the mitochondrial circuit diagram would be inherited only from women. And so, therefore, the mitochondrial DNA is transmitted from mother to all of her children and her daughters to their children. Male's mitochondria enter the egg, are seen as foreign, and are destroyed. So men have been thrown out for 2 billion years. Don't feel bad about it, guys. Okay, so the point of that is that this circuit diagram, the wiring diagram of the power plant, can only change by sequential mutation, because it cannot recombine. It never mixes two mitochondrial lineages. And why would that be? That means that any new mutation on one of the genes is tested against all the other genes of that same circuit diagram to make sure that it's compatible. And if it's not, that individual dies. So it's just like any other power plant around Los Angeles. The building of the power plants with the city managers, but the wiring diagram of every power plant is unique, and you would never think of mixing the wiring diagram half from one power plant and half from another. That would be disastrous. So the same is true for your mitochondrial DNA. Now, each cell has hundreds to thousands of these mitochondrial DNAs, and they're constantly replicating inside your cells right now. And if you doubt that they're active and vital in there, talking to each other, tonight when it's very, very quiet and your spouse is not snoring, if you just concentrate, you'll feel them wiggling in there. Anyway, they're constantly replicating, and as they're replicating DNA, they accumulate damage or mutations. And that creates then inside the cell a mixture of mutant and normal. They're called heteroplasmic. So if the cell divided down the middle, then both daughter cells would get some mutant and some normal, but if it